All right, so we just have two little I can statements here. Actually, I might mention one other one, but I can explain the role of kinases in the regulation of the cell cycle, and I can explain the role of cyclins in the regulation of the cell cycle. And so I am going to talk a little bit first about what they are. So cyclins are molecules that are created at certain points in the cycle, in the cell cycle. So if I need cyclin B during metaphase, then I only produce cyclin B during metaphase, during cell cycle. Okay, so their presence during the cell cycle is going to cycle, right? So again, if this is like P-mat interphase, P-mat interphase, P-mat interphase. Okay, so this is one that is used during mitosis, so we see it peak during PMAT and it goes away during interphase. And its activity is going to be the same. So it's only going to be active when it's present, because duh, if it's not there, it can't be active. All right, kinases. These particular kinases are cyclin-dependent kinases, okay? And we just talked about a cyclin. So cyclin-dependent kinases are only active, so let's say this is the cyclin, and here's the kinase, okay? So when it's like this, it is turned off, okay? Let me actually, where'd my red pen go? Okay, let's shade in the cyclin so we can kind of show it over here. All right, then what'll happen is it will bind to kinase to form what's called a cyclin CDK, cyclin-dependent kinase, right? CDK complex, okay? And when that happens, this turns that kinase on, okay? And if you remember, kinases are enzymes. They're enzymes that add a phosphate group to another molecule. So once the kinase is activated, it can then phosphorylate another molecule to do whatever it needs to do. This does everything from dissolving the nuclear envelope during prophase to growing the microtubules out during really all of the phases, prophase, metaphase, to activating the motor protein. I mean, literally every single tiny little step that happens to activating the cohesins to split those sister chromatids, every little tiny thing that happens in every little phase of mitosis and cytokinesis is controlled by some molecule, often a CDK. All right, the thing about kinases is they're always present. Okay, their presence never changes, because once the cyclins are produced, these guys need to already be there so they can get to work. So their presence is constant. They're always there. I probably should have shown it up here, like it's always high. But their activity is only going to be when that cyclin is produced. Okay, so if it's this same, let's say it's this cyclin we talked about over here, then we would see this CDK is only active during mitosis, because that's when that cyclin is produced and can bind to it. So when it's by itself, it's turned off. So that would be like during interphase. And then when the cyclin is created, like during mitosis, and binds to the kinase, that activates the kinase. Now, there are some that are active during interphase. So some of these would have the complete flip-flop graph. I'm just talking about one that is potentially active during mitosis. So that's the role of cyclins. They basically turn on and off kinases, and the role of kinases, which turn on and off everything else. And that's how they go. The next one is I can explain the role of internal and external signals at checkpoints. Those are just going to initiate cell signaling pathways. So external signals, like a growth factor, is going to act as a ligand that binds to a receptor. Internal signals are going to set off that signaling pathway in a different way. So it could be an intracellular receptor. It could be some other way that it can do that, but they're basically going to start either start or stop. If it's a stop signal, if you get to a checkpoint that says, no, we're not ready, the DNA wasn't copied correctly, then it'll get the signal to stop. But basically what those signals are doing is they are activating either a go pathway, where am I at? Here we go. A go pathway or a stop pathway. And those are just signaling reception, transduction, responses, signaling pathways. And so that is kinases, cyclins, and the role of internal and external factors or signals at checkpoints. I hope that helps.